Well, it really is, I'll echo some of the other comments. It really is a pleasure to have all of you here at ISB. We do science because we want to advance medicine. We want to help people that have disease, and it's really wonderful to see it all come full circle. Also want to echo others who have said thank you to Susan for bringing us together and to the Wilkies for making it possible. Really appreciate that. So I'm going to briefly talk to you a little bit about the future, where we're going, and I'll explain a little bit. So as we think about Lyme disease, and a number of people have brought this up, but especially as we think about this transition from the acute to the post-treatment or to chronic Lyme, it's very subtle and it's very complicated, as you all know, because there are many hypotheses. There's many things that could be going on, right? It could be an autoimmunity. It can be that the Borrelia gets into different parts of the body, whether it goes into the brain or the heart or wherever it goes. What this means is you have a fanning of complex symptoms that can come up in Lyme that are not obviously related to one another. And so this gives us a very complicated phenotype. In fact, I had a great conversation with one of the patients here in the audience with Nate McQueen, actually. We were just talking before. And from that conversation, we realized that you can actually even potentially have an oscillation cycle between an autoimmune type of a response. Then your body might get used to that, and now all of a sudden you have a situation where your body's no longer recognizing the Borrelia anymore. Apparently there are some stem cell therapies and things that seem to come in at that route. Anyway, we won't get into that, but it was another facet that I hadn't contemplated before, which is actually there could even be a toggling, and this seems to match up with some of the symptoms that I've heard a little bit described tonight. So all of that complexity is to say that we want to think about how do we deal with that. And Lee talked some about this. So one of the things that we've been pioneering recently is a concept that we call scientific wellness. And basically this is the notion of generating personal dense dynamic data clouds. And the idea here is to have a very broad set of measures. So when we do these, this can involve whole genome sequences for the person, and it could be proteomes, metabolomes, microbiomes, clinical labs, deep dives into the immune system, sometimes data from wearable devices and so forth. But the idea is to take a very deep dive into understanding what's happening at a systems level inside a person. We launched a study, Lee and I, a few years ago called the Pioneer 100, which took 100 people through this kind of program. And then as Lee mentioned, he and I co-founded a company called Aerovale here in town. And the reason I bring it up here is because this has taken this approach to consumers, but we now have about 4,000 people who have gone through that program. And the important thing for tonight is that every person who's gone through that program, if they check a box opting in saying that their data can be used anonymously for research, those data come back into ISB. And what that has let us do is to develop a series of computational tools for understanding these dense dynamic data clouds. And in particular, we've developed new approaches that allow us to say, for any given individual, here's every analyte in your body that's in a state that's unusual for someone who is healthy. Here's every network that's in a state in your body that's in an unusual state, given for someone who is healthy. Here are the relationships between every one of these variables that is unusual given someone that is healthy. So what this is giving us is a new approach to think about N-of-1 diagnostics, to think about how do we dive into a situation where phenotypes are very disparate and where we don't fully understand or we can't really separate into nice case control type studies. And so this approach is something that we think is applicable in areas that are particularly murky, such as post-treatment Lyme or chronic Lyme. And so this is an area that we're very excited about pursuing going forward. So what we're doing, as Lee alluded to, is we're seeking to do a project where we will get 200 individuals centered around a community that is infested with Lyme disease, and evaluate and get measures for people before they get Lyme, and then through the course of their treatment. And then we should be able to observe about 20 to 40 individuals who will go on to post-treatment Lyme. And what this will give us is the densest, most phenotyped, most studied group of individuals ever to go through that transition. And what we hope to be able to do from those data is to understand what each individual's particular trajectory looks like, what's everything that's unusual about their trajectory, and then to aggregate those together to see if we can learn something about that transition state. And so this is really a key element to everything that we're trying to put together. And we believe that in this way, we'll be able to build on the great approaches that you've heard from Neha, that you've heard from Rob, and of course that you've heard from our great leader, Lee Hood, about how we can bring this all together and we hope make really a positive difference for you and the Lyme disease community. Thank you.